processing and presentation. So when Marco came to the laboratory, the knowledge at that time how antigen is processed by immune cells and presented to T cells is summarized here in this annual review paper by Peter Creswell. So HLA-DR molecules, they are synthesized in the cell. Then in the endoplasmic reticulum, they are assembled to the heterodimer of alpha and beta chain, actually together with the invariant chain. Then these molecules are moved forward to the Golgi complex where glycosylation happens, and then they go into endocytic or lysosomal compartments where they get in contact with bacterial or viral proteins, sorry for this pointer here, that come in from the outside. So a cell takes up a viral protein, a bacterial protein, another protein by endocytosis, by phagocytosis, and then these complex proteins are digested in the endolysosome and loaded onto newly synthesized HLA-DR molecules. And these loaded molecules then go out to the surface of the cell where they're available to be recognized by a T cell. This was the state of knowledge at the time when Marco came. And in a collaboration with Eric Long, who worked in another institute around the corner, but also part of NIH, we had a very fruitful collaboration on antigen processing. Eric's long interest was primarily viral antigens. Influenza virus was their model antigen at that time. And we contributed cells and systems in the autoimmunity context. And what this work could show, that MBP, which is normally bound to lipids in our body, so lipids that form the majority of the material that makes up our myelin, 80% is lipids. When you take MBP together with myelin lipids, it doesn't need de novo synthesis of HLA molecules, but is entirely sufficient that this material is taken up, goes into the endosome, and is loaded onto HLA molecules that come in from the surface, are recycled through this compartment and go out again. So that de novo synthesis of HLA-DR molecules is not necessary for this novel processing pathway that was demonstrated by Eric Long's group and Valérie Pinet as the first author at that time. A little bit later, again now using myelin basic protein as a model antigen in another study, it was shown that a protein like MBP, which has a very high pH, it's very basic, therefore the name myelin basic protein, the pI is about 10.8. When you put it in solution, it dissolves very well, and it doesn't have a secondary structure, different from many other proteins that form beta-pleated sheets or alpha helical structures. This protein is entirely linear, available in solution. And when you add it to a tissue culture medium, what Marco could show is that the full-length protein, without any digestion, without any need to enter the cell, can be loaded onto HLA molecules on the surface of the cell, can bind there to an HLA molecule and be immediately recognized by a T cell. So that professional antigen presenting cells that are capable of doing all this here are not really necessary, that it's just required or sufficient to have the HLA-DR molecule on the surface to lead to recognition by a T cell when this entire protein here binds. It's probably then also cleaved at the surface, but it's not necessary that it is cleaved. So let me, after showing you these three different areas of Marco's work, come to just very briefly to further impact that what he did had for studies that went on after him, after he had already left back to Florence. I already showed you the study with the altered peptide ligand. I will show you now just a little bit of work that used a receptor that Marco had established, and that was one of the first human T-cell receptors that was co-crystallized with an HLA peptide complex. So what you see here in a study from Roy Mayuza's group and Yi Li Li, who was the first author of this study, is that a T-cell receptor, the TL3A6, that is 83 to 99 MBP specific, comes from an MS patient, recognizes the peptide together with HLA-DR2A, one of the two risk alleles in MS. And the interesting finding in this particular paper was that the T-cell receptor contacts the peptide different from virus-specific T-cell receptors, not by any high energy bonds. So there were no salt bridges, no hydrogen bonds, only van der Waals interactions, meaning that this interaction is very, very weak. And obviously, this raises the question, was this just some tissue culture artifact that we chased, maybe interesting for crystallization and showing that a low affinity receptor can bind to an MHC peptide complex. But a study that we are currently trying to publish shows you that such a low affinity receptor can be pathogenic. What was done in this study here is that we took the variable regions of this receptor, V alpha and V beta, and from the HLA molecule and engineered them onto the mouse constant region of the T-cell receptor and the IE region of the HLA class 2 or MHC class 2 in mice, creating all the recognition structures from the human T-cell receptor and the human HLA molecule in a transgenic mouse carrying 4 transgenes, and these animals get spontaneous disease. Meaning that this low affinity T-cell receptor, when it's put into a mouse together with the human MS-associated HLA-DR molecule, leads to onset of a spontaneous MS-like EAE in animals, showing that these receptors, even though they are very low affinity, are biologically relevant. I'm not going to go over all these papers. I just want to give you a glimpse that Marco not only did his own original work, but that he also contributed to many other studies, and even studies that are ongoing now. So there were studies on molecular mimicry with Bernhard Hemmer, studies on a different myelin epitope that Paolo mapped in the context of a different HLA-DR molecule that is also to some extent associated with MS. Studies that were focusing more on therapeutic approaches, then continuing to work with Luca here when he came back, and Benedetta Mazzanti on the lipid-bound MBP, and finally also work on signaling how autoreactive T cells receive signals and how it is transmitted into the cell. So the time that he was at NIH, the 3 years, were not only highly productive during that time, but also reached well beyond that time. So in summary, MBP and MBP-specific CD4 T cells are probably among the best examined autoantigens or autoreactive T cells for any autoantigen in human autoimmune diseases. The projects that Marco and our team also worked on are relevant, I hope I could show you, well beyond this one autoimmune disease, but also have some general implications for understanding T cell recognition in general. And we are now, many years later, going back to findings that we started to work on at that time and are now to look into in a deeper way. Let me give you, before I give you some personal memories about, or memories collected from his colleagues about Marco at the very end, some thoughts. I heard mainly students are listening to what a genuine scientist should be like, at least in my view, or the way I have been trained. So and a scientist should have a true and genuine interest in research and science, be able to ask good and relevant questions as opposed to pursuing science, which sometimes happens, only for the sake of getting a better position in academia or for other interests that are not really deeply towards science. The interest should be broad. Science is multidisciplinary now more than ever. We need to look at different techniques, so somebody needs to have an open and wide view. It is important to be persistent, not give up. Very often things do not result in nice papers. I did my first postdoc, I didn't have any paper afterwards, and everybody asked me, what did you do during that two years? I learned a lot, more than probably ever later. But what I want to say is you need to be persistent if you are interested and motivated for science. I think creativity is very important in applying novel techniques, looking for solutions beyond your immediate area and outside your direct field, able to think beyond dogma. I think immunologists are not the only species in science that are sometimes very dogmatic, and it is sometimes very hard to pursue ideas that are against dogma. I've seen this now a couple of times during my not too long career. So this is something that's important. One needs to be interested and willing to do experiments, sometimes very hard, and after a while you get also tired, like in medicine, seeing patients sometimes as well. So one needs to stay with that. And very important to me, I've got to know scientists who are not able and willing to share what they're thinking about, what they do. They're very secretive. In the environment that I grew up, it was always very important, and Marco was a prototypic example, to share ideas, to talk about them, to discuss them, and also give things to others and let them thrive with the work that you do. And as I said, Marco met this profile like very few, if anybody else, that I've met in my life. And it's really a great pity that he left us so early, 10 years ago. And let me now, at the very end, give you some memories of people who worked with Marco. And as I mentioned, Henry was my mentor, Henry McFarland. He's now retired since 1 year. He recruited Marco originally to NIH. I just let you read some of this, or maybe I read it. So Henry wrote to me when I asked him, what do you remember of Marco? And he said, one of my fondest memories was one of my first conversations with him and Marta while they were still in Florence, and that both were very concerned about their English. And Marco told me that he had been watching old American movies like John Wayne films and others every night for a few months. And indeed, when he came to NIH, his English was in fact very good. And it certainly, as an example, reflected his preparation and commitment to start and engage in this big step, I think. Bruno Gran, another colleague who came after Marco from Florence to the lab, said that Marco had a clarity of thought that I liked very much, and in science and also in life, I believe. Paolo, who came from Chieti, another very successful and good scientist, good friend, now remembered something very special that also holds true for me, is that he vividly remembers Marco saying, and he remembers it as if it were yesterday, that in his being very passionate about cellular immunology, Marco once told him about the distinction between cellular and molecular work. The thing is that T cells are alive, and when you look at them under the microscope, they speak to you. Very different from cutting pieces of DNA with a plasmid and putting them together, and this is something that's different, and I feel the same actually. Benedetta here, who is again from Florence and back here, is mentioned. I think what many others felt and feel is that Marco was a master and a true friend, and I always remember his smile, his dedication, his honesty, and love for his work and his wonderful family. And he gave me the enthusiasm for the work we do, and I remember he said that we were privileged to be able to do it. This is from Bernhard Hemmer. Sorry that it's a little bit long, but I think it brings Marco back also very well as well. Bernhard worked probably the closest with Marco during those 3 years, and he said there are many memories about Marco. He was not only a great scientist and a very close friend, but also an excellent and patient teacher, which was also very important for me, because during that time I had a double appointment in Germany and at NIH, and I went back and forth in 2-month intervals, so very often I was not in the laboratory. And Marco was really the one who held the fort and was teaching Bernhard, who was then taking onward from there. We always worked together, helping each other and sharing work and success. During our time at NIH, Marco and I went every day together for lunch and for a coffee break in the afternoon. This was always followed by a short walk around Building 10, the biggest building on the NIH campus, because Marcus had to take his cigarette. We always used this time for discussions, and I really regret not having him told him more often to stop smoking, because that was probably one reason why he finally died. But like Bernhard, I remember this very vividly, and these were the times where we discussed our work, where we had new ideas. They were not in the laboratory or not in the library. They were usually over coffee and after lunch.